is really sour. Of course, the juice that you are drinking right now is containing citric acid.
Oh, really? Guys, I have one question. What is citric acid? Cho. I know what citric acid is. Let's discuss.
Hi everyone, so today we are going to talk about citric acid. Did you guys know what actually citric acid is? Let me explain. So, citric acid is a weak organic acid naturally found in citrus fruit such as orange, lemon, and lime. It is responsible for the characteristic sour taste of fruit juice. Citric acid is widely used as a food additive, preservative, and flavor enhancer in the food and beverage industry. Fun fact, citric acid not only gives sour taste but also help to extend shelf life and maintain freshness of the product. Why citric acid content need to be analyzed? Firstly, it is to determine the quality. The amount of citric acid will affect the taste, aroma, and overall quality of fruit juice product. Secondly, it is to ensure the consistency of flavor, which is maintaining the right level of acidity will ensure the consistent sour taste and consumer satisfaction. Thirdly, it helps to comply with food standard, which is it helps manufacturers to comply with legal regulation and industry standard for food safety and labeling. Last but not least, it helps to provide information about storage and shelf life, which is citric acid acts as a natural preservative and will affect the stability and shelf life of fruit juice. The objective of this experiment is to determine the citric acid content in fruit juice using acid-base titration. To understand how this determination is carried out, the next presenter will explain more about the principle of acid-base titration method.
So next, what is acid-base titration? Acid-base titration is a quantitative analytical method used to determine the unknown concentrations of an acid or a base by reacting it with a solution of known concentrations. In this experiment, the fruit juice or citric acid is the solution with unknown concentrations, while standard solutions, which is NaO, is the solution with known concentrations. The titration is continuous until the reaction is complete, and then we can calculate the content of citric acid. For principle of acid-base titrations, the principle is based on a neutralization reactions where an acid reacts with a base to form salt and water. For citric acid analysis, citric acid is a triprotic acid, means that it can donate 3 hydrogen ions, where sodium hydroxide is a strong base that provides hydroxide ion. This is the very important part in titrations, which is endpoint and equivalence point. Equivalence point is the theoretical point where the amount of acid and base are exactly chemically equivalent. For citric acid titrations, it means all hydrogen ions from citric acid have completely reacted with hydroxide ions from sodium hydroxide. At this point, the reaction is perfectly balanced according to the mole ratio. Next, the endpoint is the practical point observed during the experiment. It is identified by a color change of the indicator. Indicator works as a substance that changes color depending on pH. In this citric acid titrations, phenolphthalein is suitable to use because it changes color in the pH range about 8.2 to 10. This range matches the neutralization point of a strong base, which is NaO, with a weak acid, which is citric acid.
Next, we are going to see the citric acid standard solution preparation. So before we are performing the titration, a standard sodium hydroxide solution is prepared. A required amount of sodium hydroxide is dissolved in distilled water and it will be transferred into a volumetric flask. The solution is then diluted to the mark and will be mixed thoroughly to ensure a uniform concentration. And then this sodium hydroxide solution will act as the titrant to neutralize the citric acid that are present in the fruit juice sample. The fruit juice sample is prepared for the titration process. The juice is mixed well and a known of volume is measured and diluted with distilled water. Phenolphthalein indicator is added to help to identify the endpoint during the titration process. Next, I will explain the titration process for determining citric acid content using acid-base titration. So the materials used include the fruit juice sample, standardized sodium hydroxide NaO solution, phenolphthalein as an indicator, and the required laboratory apparatus such as burette, pipette, and conical flask. In this experiment, the fruit juice sample is titrated with sodium hydroxide solution. Phenolphthalein indicator is added to detect the endpoint of the reaction, which is indicated by a permanent pale pink color change. The volume of sodium hydroxide used is then recorded and used to calculate the citric acid content in the fruit juice. Next, I will explain the procedure for determining the citric acid content in fruit juice using acid-base titration. So first, the fruit juice sample is measured and it will be diluted with distilled water. The diluted sample then will be mixed with phenolphthalein indicator and it will be titrated with standardized sodium hydroxide solution until a permanent pale pink color appears. The volume of sodium hydroxide used is recorded and the titration will be repeated to obtain an accurate result. Finally, the citric acid is calculated based on the titration data. The reaction involved is the neutralization between citric acid and sodium hydroxide, and it will be producing sodium citrate and water.
Now, I will discuss the application of citric acid analysis using acid-base titration. Firstly, in the food and beverage industry, this method is used to determine the citric acid content in fruit juice and beverage. It helps manufacturers maintain the product quality and ensure that nutritional information on product labels is accurate. In addition, acid-base titration is widely used in quality control laboratories to monitor the acidity levels of product. This helps ensure consistency during the production and compliance with industry standards. Okay, let's continue about the application of citric acid. Citric acid analysis is also important in nutritional research. Researchers use this method to compare the citric acid levels in different fruit juice and study their nutritional properties. Furthermore, acid-base titration is useful in product development and storage studies. It helps monitor changes in acidity during the storage and provide the valuable information for improving product formulation, preservation method, and shelf life. Therefore, citric acid analysis plays an important role in ensuring the quality and safety of the food products. In conclusion, citric acid is an important organic acid commonly found in fruit juice and beverage. In this presentation, we have discussed how acid-base titration can be used to accurately determine the concentration of citric acid through a neutralization reaction. This method is simple, reliable, and provide accurate results, making it suitable for the routine analysis. Furthermore, citric acid analysis has many practical applications in the food industry, quality control laboratories, and nutritional studies. Overall, this topic demonstrates how the analytical chemistry is applied in everyday life to ensure the product quality, safety, and consumer satisfaction. Okay, that's all for me.